I'd like to thank Ying-Yi Sun and my friend and colleague, Professor Henry Yang, for inviting me to speak at ICG 18, which is the latest in a remarkable series of meetings, remarkable for its breadth, depth, and inclusiveness. What I'd like to speak about is implied by the title of the first slide, namely, Agricultural Genomics offers us a trifecta of opportunities. It's certainly well known that it can improve food security substantially. For example, the Gates Foundation is heavily invested in that. A little less emphasis on the possibility of discovering new drugs, which is there. But what's not discussed, hardly at all, and certainly when I got interested in this subject 5 years ago, the possibility of mitigating and adapting to climate change. And that's what I'll speak about and try to convince you that there's an opportunity for a major contribution. But there are also substantial challenges. One of them is societal, two broad categories. And in the societal category are regulatory impediments, especially since we're going to be talking about using biotechnology, and there have been major obstacles, regulatory obstacles, to introducing it into the food business. And the other, which is maybe in fact more substantial, is international cooperation. Obviously, climate change is a global problem. It's not going to be solved by any single country or any single set of countries. Cooperation is absolutely essential, and the scientific community is going to have to start taking some concrete steps to move politicians in the right direction. The other set of problems are scientific and engineering. The ocean-atmosphere terrestrial system has multiple and complicated positive and negative feedback loops. They're nonlinear. It's a very, very difficult system to understand what happens if you perturb any particular part of it. Then there are the engineering problems, developing reliable, cost-effective alternatives to fossil fuels. That's receiving a lot of attention. At least there's a lot of talk about it. In terms of implementation and cost effectiveness, that's another problem. Then there's the part we'll be speaking about, removing greenhouse gases from the atmosphere. And I'm taking as a given that greenhouse gases produced by human activity are the major contribution to the climate change we're experiencing. When I became interested in this subject about 5 years ago, the information on the next couple of slides caught my attention and suggested that emphasis on regulation alone wasn't going to solve the problem. And I have to add that my point of view was in the distinct minority, and maybe still is. Slide 3 shows that approximately 28% of a pulse of CO2 emitted today will still be in the atmosphere after 500 years. In other words, within the next 1 or 2 or 3 or 4 generations, CO2 enters the atmosphere and it's there essentially irreversibly. Secondly, approximately 50 gigatons, 50 billion tons of CO2 equivalent enters the atmosphere annually. And currently there are more than 1 trillion tons of excess CO2 in the atmosphere and almost 1.5 trillion tons of CO2 equivalents. By excess CO2 or CO2e, I mean the difference between the current CO2 levels and the pre-industrial levels, more or less the 1850 to 1900 average. For those of you who may not be familiar with the concept, a CO2 equivalent is the amount of a greenhouse gas that produces the same global warming as some given concentration of carbon dioxide. So by working in CO2 equivalents, we can simply tally the CO2 equivalent of each greenhouse gas and get a number that makes some sense, that's properly normalized. I have to say there are some difficulties, at least I have some difficulties with the concept because warming does not mean temperature in this definition, but I'm going to ignore that and do what everybody else does and talk in terms of CO2 equivalents. What's being done about this? Well, the United States recently passed a bill committing some $400 billion to climate change mitigation. And by mitigation, they mean mostly looking for non-fossil fuel alternatives. The implications of the bill have been analyzed in detail, and it's expected to reduce greenhouse gas emissions from between 32 to 40% below 2005 levels by the end of the decade. That's the claim. The question is specifically, how would the temperature differ from what it would be if the world continued along its current business as usual trajectory? So we know what the target for CO2 levels are, and we know how things are going right now. And what we'd like to do is project the difference between those two scenarios and see how much of a difference this makes. And since we're talking about global temperature, we're going to have to assume that every country does the same thing that the United States is doing. So that's obviously very, very unlikely, but let's assume it. We'll get a best case scenario. In order to project the temperature difference, we need several types of data. One is the global CO2 emission. If we continue with business as usual, how is CO2 emission changing? How has it changed over the past 20 or 30 years? It happens, I'll show you that result in a minute. The annual rate at which CO2e decreases in order to meet a future target. The fraction of CO2e emission that remains in the atmosphere, and an empirical relationship based on historical data between atmospheric CO2e and average global temperature. As shown on the next 3 slides, the relationships are linear, and because of that linearity, we can make the projections. If they weren't linear, we wouldn't be able to do anything about projecting. So we determine the parameters by best least square fits. We throw it all into an algebraic hopper, and we get an answer and we get an expected difference in atmospheric concentration of about 1%. So assuming the whole world cooperates in this, we'll get a difference of about 1% over the next decade, and that's sort of insignificant. It becomes worse when we translate that into temperature. That is, we use the relationship between temperature and CO2. The temperature difference is about 4/100ths of a degree centigrade. The reason for that is twofold. One is that, as I said, when CO2 gets into the atmosphere on any reasonable time scale, and certainly in the next decade, it's there basically irreversibly. And secondly, even though we're reducing the amount of CO2 emitted, we're still emitting it, and whatever we emit stays. The result is that this strategy, which focuses on regulation or switching to alternative fuels, has essentially no effect, even if it's done globally, worldwide. What's missing is the plan has to be more ambitious with respect to a search for alternatives to fossil fuels, but also we have to worry about getting carbon dioxide out of the atmosphere to get around this problem, the stickiness problem. In terms of what to do about it, the philosophy here is that climate change is viewed as a land management problem. Increased crop yield, soil carbon sequestration, and more efficient use of land means increased drawdown of carbon dioxide. The connection has been recognized for decades, but genomics, and this is more recent, genomics augments and amplifies traditional methods for improving crop yield, but it also introduces entirely new methods, and I'll get to that in just a few minutes. Slide 13 shows the results of a model which predicts actually carbon dioxide in the atmosphere during the course of the current century. The upper curve shows what happens if carbon dioxide emissions are brought to zero continuously, linearly, over the course of the century. And you can see it continues to increase because even though it's being brought to zero by the end of the century, it's still increasing. And the result is about half the amount that would have gone into the atmosphere goes into the atmosphere. The 2 lower curves shows what happens under the same condition, but now using engineered trees with different efficiencies. The diamonds show what happens if we use only engineered trees, and the black curve shows the effect of a 20-year delay. The 2 main messages here are that, one, avoiding a delay is essential. It has a huge impact. A delay has a huge impact, and we are delaying. And the second is that neither strategy by itself is sufficient. Even if we engineer trees and we don't regulate carbon, that's liable not to buy us very much. It's going to keep us where we are. And of course, if we don't engineer the trees, carbon in the atmosphere is going to continue to go up. Let me move on to some concrete examples. This comes out of Julie Gray's lab at the University of Sheffield, cellular engineering. Stomata, these microscopic pores in rice and other plants, regulate carbon dioxide uptake while releasing water vapor. Elevated temperatures, carbon dioxide, and drought trigger stomatal closure. That's how the plants regulate themselves. The Gray Lab has engineered a low stomatal density rice cultivar by manipulating a developmental signal with reduced water release and increased drought and disease tolerance. This can also be done with other plants, and it has clear implications for adaptation of crops, in particular crops resistant to changes in climate. And also, it certainly allows the possibility, at least, for growing on land which is currently considered non-arable. Another example is replacing synthetic fertilizers. Fertilizer produced by the Haber-Bosch process accounts for about 2% of greenhouse gas emissions, both in producing ammonia and in also transporting it. And about 50% of the synthetic fertilizer applied to crops runs off into groundwater and create dead zones. That's because the crops take up this fertilizer very inefficiently, and so a lot more has to be applied than is necessary. There are a number of potential bioengineering solutions. A company called Pivot Bio has engineered microbes to produce nitrogen fertilizer from atmospheric nitrogen. Anuvia Plant Nutrients developed an organic matrix that releases attached nutrients, avoiding runoff and the use of synthetic fertilizer. And then there are also soil-free systems, not really biotech, but certainly important and worth looking at. Finally, let me say a few words about photosynthesis, which is where a major impact can be had, but the problems are also staggeringly difficult, but solvable. One of the fundamental facts about photosynthesis is that it's very inefficient, and the origin of the inefficiency is the rubisco molecule, the most abundant enzyme on the planet. Rubisco catalyzes two reactions utilizing the same reaction site. It catalyzes carbon dioxide and it catalyzes oxygen. When it catalyzes CO2, it's doing the right thing. When it catalyzes oxygen, it leads to a different pathway which is toxic. The takeaway from the next slide is some work being done in the laboratory of Tobias Erb. This is slide 18. And they're in the process of engineering more efficient photosynthesis pathways and trying to actually engineer entirely new photosynthesis pathways. I also think it should be possible to engineer more efficient rubisco. Now, not everyone agrees that there'll be much payoff in that for some technical reasons that I won't go into. But I believe not only is it possible, but it could make a substantial contribution. We're looking at this and we're using AI in many aspects of it. Finally, just to remind everyone, there's a third arm to this trifecta, and that is medicine. And currently, some 11% of the drugs classified as essential by the WHO originated in plants. Plants have been traditionally a very important source of drugs. With the ascendancy of structural biology, during the past several decades, there's been reduced emphasis on plant-based origins, but in fact, these chemical and structural methods have not so much displaced natural products, which is what I'll call them, as a source of drugs, but they've accelerated the opportunities for exploiting plants in the search for new medicines. So sequencing enables rapid, inexpensive elucidation of structure and the structure of enzymes involved in the synthesis. Design enables increased potency, and low-cost synthesis enables amplification of availability. And an example of this is the Vica alkaloids that are widely used to treat various cancers. But the general idea should be clear. The plant pathways provide the template, and then the use of chemical and structural methods are used for optimization. Here's a very brief summary. A massive agrogenomic project, which is what I have in mind. If it's done that way, there could be some payoffs. It's applied to climate change and could result in a triple win: increased food security, reduced atmospheric carbon, and discovery of new classes of pharmaceuticals. But it's got to be really extensive, and it'll dwarf the Genome Project if it's done, for sure, in terms of payoff. The main obstacle to progress will not be scientific, I don't think. I think it'll be societal, and that includes regulatory impediments to genetically modified plants and bacteria and international cooperation. Thank you all very much.